Hello, this is Michelle Miller. In the last video, we talked about mitosis as a type of cell division where you produce two daughter cells from one parent cell, and those cells are genetically identical to the parent cell. So this would be the stages of mitosis. You can ignore this pro-metaphase, so you can just go from prophase to metaphase, anaphase, telophase, and the very end is the cytokinesis. We also talked about how this was regulated. So the mitosis is highly regulated. So we only want cells to divide when they should, and we want them to not divide when they shouldn't. So we talked about the cell cycle and how there are internal regulations, including the presence of a protein that's called cyclin. And there are different checkpoints where there has to be a certain level of cyclin present and a certain type of cyclin present in order for the cell to continue. So a checkpoint is an area where the cell could stop if it is not ready for mitosis. We also talked about the mitotic clock, which has to do with the telomeres and the amount of telomere material that is at the end of the chromosomes. And remember that this specifically deals with regulating how many times a cell can reproduce. So if you lose all of the telomeres from your chromosomes, then the cell will not be able to reproduce. We also need to talk about apoptosis. So apoptosis is kind of the opposite of mitosis, wherein this is actually an example of programmed cell death. So cells need to know when to reproduce, but they also need to know when to die. And there are certain times in our life where we have lots of cell death, and that is in embryonic development. So in embryonic development, certain structures appear and then they have to disappear. So the kidney is rebuilt, as would be the tissues that are between the fingers would have to actually undergo apoptosis in order for the fingers to appear. So that webbing between the fingers would be an example of apoptosis, programmed cell death during embryonic development. So this sometimes occurs in embryonic development. Now notice that this is programmed, so it's not just cells dying haphazardly or being killed. So this means that the cell actually is the one that initiates its own death. So you could almost think of this as like cell suicide. So another place where apoptosis is important is when cells have become damaged and could possibly become a danger to the body. So we could say damaged cells. A good example of this is what happens when you get a sunburn, because excessive UV radiation can damage the genetic material inside the cell. And we'll talk about exactly how it does this when we start talking about mutations. But if the genetic material if the cell senses that it has been damaged to the point where the cell could actually potentially become cancerous, then the cell has to undergo apoptosis. And so this is just an example of this, but damaged cells die to protect the entire body. So it's a protective measure in that particular instance. Now this directly relates to a problem that sometimes occurs when cells reproduce in an unregulated fashion. So we call this cancer, where cells have unregulated cell division. And it's kind of interesting when we think about our bodies, we have all of these somatic cells in our body that are only surviving to make sure that the cells that are in the ovaries and the testes, the ones that are going to become the gametes, can survive and can be produced. And those are the cells, the germline cells, are the ones that can potentially be passed from one generation to the next. And so our somatic cells actually have to cooperate in order for that to happen. So it's not that they can just reproduce whenever they want to. So when we look at cancer, we can look at the characteristics of cells that are cancerous. And one thing that we see is that the cells that become cancerous become unspecialized. So say, for example, you can have a specialized cell in the breast, and it could become unspecialized, so it could live anywhere in the body. So rather than living in one particular location, it could end up in the lungs, in the liver, in the brain, anywhere in the body. And so this allows the cells to live anywhere in the body. And when you look at these cells, they can look abnormal. And one way that they look abnormal is that they tend to have abnormal chromosomes. And they can also have abnormal nuclei. So when we look at certain cancer cells, let's see if I can find my cancer cell video. Oh, I forgot to put that image on there. Okay, never mind. So we're talking about cancer. Okay, so for example, when you get a pap smear, when females get a pap smear, they remove some cervical cells and they look at them underneath a microscope and they stain them, and they can actually tell if the cells are becoming precancerous or if they are already cancerous. And so the whole reason for pap smears is to detect or to look for cancer cells of the cervix. Now, the interesting thing about that is that particular type of cancer is due to a viral infection that's called the human papilloma virus, which attacks cervical cells and other reproductive cells. And it can also cause genital warts. But that virus can actually damage the genetic material and cause these cells, the normal cervical cells, to become cancerous. Okay. So the third thing is that they are able to reproduce in crowded environments. And so we say that they have no contact inhibition. So what that means is that normal cells, if they are close in contact with other cells, or if they sense that there's lots of waste material being produced by other cells, they will normally stop dividing. But cancer cells will continue to divide and they will form tumors. So this is the formation of tumors. Now, not all tumors are cancerous, right? So specifically malignant tumors, and this is versus benign tumors. So malignant tumors are cancerous. And so if you do a biopsy and they look at the cells in the tumor and the cells look funny and their genetic material is all messed up and the chromosomes are like broken and the nuclei are like huge, then they will tell you that you have a cancerous tumor versus a benign tumor. Okay. The fourth thing is that they do not undergo or don't undergo apoptosis. So they do not commit suicide to protect the entire organism. The fifth thing is that they have no mitotic clock. And we talked about how the telomeres can actually lengthen under certain circumstances or shorten under certain circumstances. And it appears that it is due to an enzyme that is called telomerase. So telomerase adds the telomeres back onto the chromosomes. Okay. So in certain circumstances, telomerase is produced, but it is produced by cancer cells. And so what this means is that they can divide indefinitely. So they're essentially immortal. So they could go on dividing over and over and over and over again, and they will never stop dividing. So they are immortal. Now, the cells, because they're unspecialized, can then also enter into the circulation. So they can enter the circulatory system. And when they are in the circulatory system, they can go anywhere in the body. And this is called metastasis. Right, so if your cancer has metastasized, that means that it has entered the circulatory system. So even if you remove the tumor and you radiate that particular area, you still are under potential danger of having cancer just pop up anywhere. And so that's the worst part. And so generally that has to be treated with chemotherapy, because the chemicals put into the circulatory system will go everywhere in the body, and they will kind of shut down all rapidly dividing cells, which is why cells that are producing your hair die, and so the hair falls out, for example. So that would be another example of a characteristic of cancer cells. Now, what we know about cancer cells, a lot of what we know about cancer cells has to do with what are called HeLa cells. And HeLa cells are kind of famous now because there was a book that was recently published that is called The Immortal Life of Henrietta Lacks. And it's a great read. Specifically, it's nonfiction, but that's the L-A-C-K-S. It's a great read because it talks about the way that medicine was performed in the 1950s. And so it's very interesting. It's very different than we did today. It is very highly controversial because HeLa cells were taken from a woman named Henrietta Lacks without her knowledge. So it's a specific group of cells that were taken from a single woman. And the cells were actually cervical cancer cells. And they were in the 1950s, they were taken. And then these were the first cells that we could really, or scientists could really have reproduce in a test tube. And so they were so excited about it. And they were also one of the first commercially available lines of cells. And so even today you can buy, if you're a certain university or a research company, you can buy HeLa cells, and these cells can be used to do research. The most interesting thing about them is that they are super resilient. And so without people realizing it, and not super resilient, they're super resilient. And without people realizing it, the cancer cells actually took over whole labs. So HeLa cells invaded normal cell lines in labs all over the world. So what they discovered is that even like if you wash your glassware, there could still be HeLa cells adhering to it. And then they could transfer to another Petri dish or another test tube, and they would just outcompete the normal cells. And so it was really interesting because people had done all of this research on what they thought was normal cells. Like I could be studying what I thought were normal pancreas cells. And then when they discovered that they were taking over the world, then they had to go in and look at all of everybody's cells, and they're like, oh my goodness, this is not what I thought it was. These are actually HeLa cells. And so they actually had to go back in and rewrite all of their research to add that caveat that these were actually HeLa cells and not regular cells. Okay. And then obviously the moral issue is whether or not these cells should have been taken from a woman without her consent. And she was in her 30s, and she was Black, and she was poor. And so her family are really kind of upset today about the use of her cells without their previous knowledge. But HeLa cells are really pivotal, or were really pivotal, and are still really pivotal in our understanding of all sorts of things related to cancer, but also related to just cells in general. So if we look at the genetics of cancer, we haven't talked about genes, but in your book, they talk about the genetics this time. And so I'm just going to introduce the concept of a gene. So genes are pieces of DNA that code for proteins. And we actually have variations on genes, which are called alleles. So alleles are variants of a gene. So variation. So we're going to talk about dominant alleles and recessive alleles. This might be something that you have learned about in previous biology classes because it's usually discussed. But in this particular instance, we're going to take a look at two different categories of genes. And the first one are called proto, and I'm going to put oncogenes. Oncogenes. And an oncogene is a variation, an allele, that leads to cancer. And oncogenes specifically, and proto-oncogenes specifically, produce, they activate cancer. And so they actually cause, instead of suppressing cell division, they actually activate cell division. So they enhance cell division. And this type of cell division we're still talking about is mitosis. So these enhance cell division. So the proteins, right? The protein enhances cell division. So when you look at this word, proto means before, right? So we all have proto-oncogenes. Right? But those proto-oncogenes could change, they could mutate, and they could form oncogenes, right? So, and if they mutate or change, they can become oncogenes. Okay. So one example of this is the gene that codes for the most aggressive form of breast cancer, which is called BRCA1. Right? So BRCA1 is a gene that causes the type of breast cancer wherein females will actually have their breasts removed and their reproductive organs removed because if they're so likely to get it that they want to protect themselves from it. So it runs in the family and it's deadly. Okay. The other type, so we have proto-oncogenes. We also have what are called tumor suppressor genes. And these are proteins, or these genes code for proteins that inhibit cell division, mitosis. And our most important one is called P53. And P53 is a super important one. Now we have two variations of this. So I could just write P53 and maybe I'll put an A here, P53A, okay? So this is the normal allele. Now P53, we all have it, but it could be the mutant allele is B. So this is the mutant allele that causes cancer. So it produces a defective protein that cannot regulate cell division. So this is produces a defective protein. Okay, so sometimes we say cancer runs in the family. We all have genes that could mutate and become cancerous. So we each have 2 copies of these. So if I have P53A, and I have another copy that I get from my mother, which is P53A, right? Then I have low risk of cancers that are caused by that. Because in order for me to get cancer, both of these would have to change to P53B, right? If I have 1 copy of the mutant allele, P53A, right? So if I have 1 copy of the mutant allele, this means that I am at high risk. And then if I have 2 copies, this means that I will get cancer, right? I'm for sure going to get cancer. So if we look at cancer cells, they will have 2 of these mutated. So this is cancer, right? And so when we say that you have inherited the risk, a high risk of getting cancer, it's because you have inherited 1 bad allele, but you still have 1 good allele. So what you do is going to affect whether this one changes. So if you smoke, then this one could change. If you eat preserved meats, then this one could change, right? If you don't exercise, then this one could change. And so there's things that you can do in your environment that are going to increase the likelihood of this one mutating, right, and producing another bad allele. So in your book, they talk about this. Okay. So this is the normal P53, and it causes apoptosis if the cell is damaged, right? And so it inhibits, it causes the cell cycle to arrest, and then it will also help to repair the DNA, and then it'll restart the cell cycle. So mutated means that even if the cell is damaged, it is not going to stop reproducing. And so it will continue to reproduce. And so then you get an increase in the likelihood of developing cancer. P53 is responsible for almost, I think somebody estimated 50% of the cancers, including colon cancer and lung cancer. Okay, so we're going to next talk about meiosis. And so meiosis only occurs in the ovaries and testes. It only occurs in germline cells. And early during embryonic development, these cells that are destined to become egg and sperm actually seed, they call them seeding, the ovaries and the testes. So it's really interesting from that point of view, which cells get chosen to be potentially passed on to the next generation early on in a person's life. Okay, the other thing about meiosis that's important to realize is that it produces haploid cells. So haploid cells have half of the genetic material, right? These are also called gametes, right? So the egg and the sperm are gametes. And this in humans means that we only have 23 total number of chromosomes. And so there are no pairs in a haploid cell. So to produce a haploid cell, you have to actually have the one cell that's going to give rise to the gametes divide twice. And so it involves 2 consecutive cell divisions. And these are called meiosis 1 and meiosis 2. Meiosis 1 has all of the stages that mitosis has, and meiosis 2 also has all of the stages. And so like if we were to look at this, it would also have prophase 1, right? Metaphase 1, anaphase 1, telophase 1. This one would have prophase 2, metaphase 2, anaphase 2, telophase 2, okay? And that's important because anytime you see a 1, Roman numeral 1 or 2, then I know that I'm talking about meiosis, not mitosis, because mitosis has only one cell division. It only divides once. Okay. So if we look at this process, we have one cell, this is the germline cell, and it is actually diploid. And it is going to divide once, okay, but it's not through, and it's going to divide again. So potentially from one diploid cell, because this is exponential growth, you could potentially have 4 haploid cells. So these are 4 haploid cells. Right? So this would be meiosis 1, and this is meiosis 2. Now, if we look at meiosis, the cells that are produced are not genetically identical to the parent cell, and so this is what is referred to as sexual reproduction. And in sexual reproduction, we have recombination. So we have recombination, recombining of the genetic material. So it is different than cloning, right? And it's different than mitosis, where the daughter cells are identical to the one parent cell. So when we look at sexual reproduction, there's a couple of things that happen during meiosis that do not happen during mitosis that are important because they actually increase the variation in the genetic material in the haploid cells. And so we're going to look at those processes next. Okay. So the first process that happens is called prophase 1. Or it happens in prophase 1, sorry, and it is called crossing over. And this does not happen in mitosis, but here you can see the replicated pairs of chromosomes. These are the genes, and these are the alleles. So this gene A has 2 forms, a capital, which would be dominant, and a lowercase, which is recessive, okay? So crossing over is the exchange of genetic material between homologous, replicated, these chromosomes are replicated, chromosomes. Now they're homologous because they have the same genes, right? And they're the same size and the same shape, but they both have gene A, right? So this one, if it had gene E, D, and F, it would not be homologous, okay? So what happens here is that you notice, voila. So after prophase 1, you start to see these chromosomes that in diagrams, labeled red and blue, are kind of mixed. And so here you can see that this one no longer has A, big B, and big C, but now it has big A, big B, and little c. And that is more variation than we would see if crossing over did not take place. Now, here we're just showing crossing over of the genetic material once, but crossing over, it's almost like if you had a group of blue playing cards and you had a group of red playing cards and you shuffled them. So there's a lot more exchange taking place, and it is actually kind of random exchange of genetic material. And so one thing that this can cause is there's a cost to this because it could cause, so crossing over can cause mutations, right? Sometimes it even also causes deletions of pieces of chromosomes if they don't get switched over properly. But we'll talk about chromosomes a little bit later. So this is the cost, right? The benefit is that it increases the genetic variation found in the gametes. And we believe that genetic variation is super important because it, from a population point of view, allows populations to survive things like viral infections or bad bacterial infections, or maybe even parasites, right? In the past, it was different. It could even, predation could have been a problem, but our biggest risk now is from viruses and other microbes, okay? So that's the benefit, is it increases variation, but there's a cost to that, okay? Now, once prophase has happened, and that is where the chromosomes condense, right? And then crossing over happens. Then in metaphase 1, we have the lining up of the replicated chromosomes as pairs. So I'm just going to go metaphase 1. We have pairs of replicated chromosomes lining up in the center. Right? So that's different, because in mitosis, it's just single replicated chromosomes. So pairs would look like this. So there's my blue. That one generally means that it's from the father. And this is my red, right? And so in anaphase 1, what is going to happen is that these chromosomes are going to be pulled apart. So they're pulled apart like this, right? And so the resulting cells after meiosis 1 are still going to have replicated chromosomes, because the chromosomes were not pulled apart, as is in mitosis. And so that makes sense, because in this next subsequent cell division, meiosis 2, that is where the replicated chromosomes get pulled apart, and then we get unreplicated chromosomes being produced. Okay, so it's important to be able to identify the difference between metaphase 1 and metaphase 2. So if I drew my metaphase 2 here, right, I would have my single chromosome, right, lining up in the center of the cell. And then these are pulled to opposite ends, right? Oops. And so, oh, I should have done a different chromosome here, sorry. We'll make it different size and shape. Let's use red. Okay, so this could be a different, oops, let's use blue. No, we need red. Okay, so this could be a different shaped chromosome. So let's say that it's this one that's got really long arm and really short arm. Okay, and so the resulting cells, I kind of messed that one up. The resulting cells would actually have one copy of this chromosome and one copy of this chromosome, and they would not be homologous. Okay, so in your book, they have pictures of these. Metaphase 1, they're lined up like this. Metaphase 2, so this is where I kind of screwed up on the last one, but you can see how these are not homologous pairs anymore. And then we get the production of gametes, which have only one copy of each chromosome. Okay. Now, the way that the chromosomes line up also increase the variation. So they're just showing 2 pairs here, but remember that we have 23 pairs, right? So the way that they line up with like father, mother, mother, father, blah, 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 you know, is extremely variable. And so that way, the way they line up is going to determine what chromosomes end up together in the resulting gametes. Okay, so by the end of meiosis 2, we have unreplicated chromosomes and haploid cells. Okay. So this just shows that crossing over, right? And it also shows a chromosome where you have recombination has occurred, right? And then having it pulled apart. Okay, so the last thing I want to talk about is the formation of sperm and eggs, and these together are called gametogenesis. So think gamete production, gametogenesis. So let's look at the spermatogenesis first. So when we look at the cells that are going to give rise, the germline cells, they are going to eventually produce from one diploid cell, just like we would predict, 4 sperm. Okay, so I'll just draw my sperm here. And they actually are smaller than this because they actually lose a lot of their cytoplasm. If this is the nucleus, it's in the head of the sperm. Oops, there's a C there. We'll just ignore that. Okay, we'll just make that a nucleus and a nucleus. Okay, so these are the sperm. So spermatogenesis is we start out with one diploid cell in the testes, and it undergoes meiosis 1 and meiosis 2, and we produce 4 sperm. Okay. Now, when we divide up the cytoplasm, we have what is called equal cytokinesis, which means that each nucleus gets the same amount of cytoplasm, and the cytoplasm has all of the organelles in it. Now, in contrast, we'll look at oogenesis, and this is the production of eggs in the ovary. So we start out with one diploid cell, and it divides, but only one nuclei gets all of the cytoplasm. The other nuclei just gets a tiny little bit of cytoplasm. Okay, so that's at the end of meiosis 1. Generally, this doesn't divide, but it might. And then this one divides, that's meiosis 2, and it would produce one little nuclei and then one big cell with a nucleus. So we would say that this is unequal cytokinesis. So it's the unequal distribution of cytoplasm. These right here are called polar bodies. And the reason why they're called polar bodies is that they actually adhere to the outside of the egg. So they're like right here and right here, right? When the egg gets ovulated. But polar bodies are just nuclei with very little cytoplasm, and they have no function. So when we look at this, the whole reason why we have unequal cytokinesis in the production of polar bodies is that we need to produce an egg cell that is large, that has all of the nutrients that the embryo is going to need early on to undergo mitosis. So right after the egg fertilizes, it's going to start dividing mitotically and separating that cytoplasm. And it's not until it gets down to the uterus where it's going to start to use some of the nutrients that are secreted by the uterine wall. And then the little embryo, multicellular embryo, will embed itself and digest its way into the uterine lining to get nutrients. Okay, so oogenesis is the production of egg through unequal cytokinesis. We start out with one diploid cell, and we only get one egg cell. Okay, so that is it for cell reproduction. The next topic is going to be Mendelian pattern of inheritance. And this idea of meiosis is going to be really important to understanding how genes separate and then come back together, and you can use it to determine the probability of getting a particular genotype in an offspring.